So I'm going to start off by asking you, how many of you have any idea what is going on? Day of the Triffids, hands up. Who's heard of Day of the Triffids? Ah, how many of you had to go and Google it? Sort of, what on earth did you not? Maybe German, maybe it was a British thing or something. Okay, so when I started off, when I was doing my PhD, I really got into the sort of science communication. And I went on numerous sort of NERC communication training events, and also I took part in a couple of, it's called the British Association for the Advancement of Science. I went to 2 of their annual sort of conferences to present the work of my PhD. And time again they were saying, you've got to come up with a catchy title. You've got to get that hook, you know, to get people in. So I'm really glad that I've managed to fill most of the chairs tonight with this Day of the Triffids. And I'll go tell you a little bit more about it. But just to embarrass myself further, that is just a picture of me during my PhD with this. I created this out of paper mache and chicken wire and bamboo. Anushka's nodding there, she saw this for real. And I probably delivered a lecture, a lecture about my PhD to over 5,000 children during that 3 years, and because at various Shell, the oil company Shell, organised lots of education events. So I was very much involved in the science communication side as well as doing my academic studies. But for those of you who are not aware of Day of the Triffids, I will just go through it very briefly because I've tried to make my talk align with this. Don't ask me why. I just had this sudden desire to do this. Anyway, so there was a novel. Who's read the novel? Oh my gosh, excellent. Okay, then it went into a science fiction film, which, no, maybe. And then it was the TV series that got me hooked, and it really scared me. Sorry, boys, but I'm not going to, you're not seeing it just yet. But oh my gosh, it was scary. So for those of you who don't know what it was all about, I'll just have a brief recap. So Bill Masson, scientist, worked on a Triffid farm. These were these huge plants. They were mobile, they were carnivorous, but they were fantastic at producing this plant oil. So that's why they were grown on this farm. Meteor shower hit. Whole of mankind was turned blind, apart from Bill and a few others. I can tell you're hooked now, I can see this. But then after that, Triffids escape from the farm, run amok, and go around pretty much killing all the blind sort of population. And that's when it got really scary, and I think that's when I started having nightmares, but anyway. Running amok, Bill and partner provided a protective fence around the family so the Triffids couldn't get into harm them, to eat them, to kill them. And then after a while, they obviously learned that there was another sort of small population living on an island that they decided to go and join. It was obviously getting a bit too boring, or, so they decided to go and join them. And they found a way of killing the Triffids. And can anyone remember the key to killing those Triffids? Seawater, salt water, right? This is really key because we'll come back to this. I'm going to quiz you on this a little bit later, all right? Quiz you on these things. So, okay, that's just a background. And you'll notice that I've put in italics some of those words there. So I'm going to kind of use them throughout my talk, all right? Tenuously. So I've never worked on killing, any plants that go around feeding on human beings. Okay, I'd just like to make that clear at the start. But maybe you'll see something as we go through. So I just wanted, though, to give you some background as to how it all began and why I am standing here today. And Mum will kill me here. Here we go. There's my mum. My dad, unfortunately, couldn't make it today. But, so when I was very little, Mum was a biology teacher, and she always used to drag me along to her field trips in this beautiful parkland that we had right on our doorstep in Birmingham called Sutton Park. So I was always dipping for sticklebacks and various things and chasing the dragonflies while Mum was trying to do quadrat samples with her students back then. So that's sort of where my love of nature really came from. So I have it all to owe to my mum. But then during my degree, I went to Bristol to do my degree. I had the fantastic opportunity of going on a sort of conservation expedition to Belize, with Coral Key Conservation, where I met some fantastic friends. This guy here, Simon Williams, he couldn't make it today, but he'll kill me if he knew I was showing this picture. Maeve knows him. So I'll come back to Simon a little bit later, but as part of my undergrad as well, I also went to the Scilly Isles and took part. I was a great diver, scuba diver. I did some survey work on seagrass beds in the Scilly Isles, and then I went back to Belize for a second time as the scientific officer with Coral Key. So it really became embedded. I loved, every other minute I was in the water diving, and I just couldn't get enough of the water. And I also did a gap year when I travelled as well to Australia and Indonesia diving. So I was very, very fortunate. And that's where, as I say, my love of biology sprung from. I should also point out we've got little clapperboards up here, so sort of act one, following the Triffid theme. So, okay, so back to the, cultivating the Triffids, or in my case, it was cultivating the sea urchins. And I cannot say thank you enough to Maeve, who managed to come here, Maeve, who was my PhD supervisor. And really over those sort of PhD years and onto my postdocs, we were looking at basically how we can grow the most nutritious sea urchins that you can find. How can we grow them the fastest? How can we look at growing their roe, their gonads inside here as well? Because this is what people eat. I don't know how many of you have ever eaten sea urchins, but they're very tasty. We just don't eat many of them here in the UK, but they're gorged on in other parts of the world. And I looked at very much looking at growing them with a whole variety of things. And you'd never think of, you think of sea urchins feeding on kelp. But it just turned out that Maeve, there was a salmon cage, wasn't it, that was brought ashore, and it was full of urchins. And they were very full of roe. So we thought, okay, we'll try them on salmon feed. And as you can see here, this darker bar, you can see the difference in the size of their roe when they were fed on salmon feed compared to good old just sort of kelp down here. So we were like, wow, we're onto a bit of a winner here. Okay, let's take a look at how fast the whole shell grows. And that didn't do too badly either. Feeding them on salmon pellets or the salmon feed, they did actually pretty well here too, compared to sort of just feeding them on kelp. So we carried on. And okay, right, you can grow them pretty fast. Think of those Triffids, right? We can grow those sea urchins pretty fast. We can fill them up with a good quantity of roe that people are going to want to eat. But how nutritious are they? Are people really going to benefit from eating these? So we looked at in detail at their fat composition. They're called the sort of fatty acids, those essential fatty acids. We looked at that composition on their different diets. We also started to look closely at a particular type of macroalgae called Palmaria palmata, which is commonly called dulse. In Ireland, they eat it over there, but it has a very, very high protein content, can be up to 40%. So we were like, aha, because the salmon feed had quite high protein as well. So we thought, right, well, we could possibly feed this to the urchins, not be so reliant on the salmon feed, and they would grow pretty well, and they didn't do too badly. So we were like, okay, we're doing all right here with cultivating them. And it wasn't just the actual adult sea urchins that we were interested in as well. So Maeve was in contact with a wonderful lady called Wei, who worked in China at the Yellow Seas Fisheries Research Institute. And she came over here for a year and brought her daughter, who was about 12 at the time. I think she went to Oban High School and sort of learned English. And it was incredible that she brought her over, and they stayed, and Wei worked. Oh my gosh, she got 3 papers out of a year's work here, and she studied the larvae, the urchin larvae, and sort of looked at sort of different types of feed, again, that you could feed them on, so we could try to reduce that cultivation time. So we could get the larvae through to what's known as metamorphosis, where they basically settle out onto the seabed and almost flip inside out and become these miniature urchins. And when they metamorphose and settle out, they're only like a millimetre in diameter. They're tiny. But she sort of tested them out on sort of a standard diet here, and here you see a nice sort of robust, good-looking sort of sea urchin larvae, and then we tried them on this, called it micro-encapsulated feed, because it's a bit of a faff growing up the microalgae, and whether you could find some formulated diets to grow them on would help in a cultivation scenario. So we saw a little bit of stunted sort of leg growth, but not too bad. But then we also tried, or Wei also tried, growing them on this sort of algal paste, which is commonly given to shellfish. And as you can see, they didn't really do too well on that. So, but it was a really interesting study, and it was fantastic working with Wei. So part of this as well, we weren't just interested in the actual growing of the urchins themselves, but this was work by Adam. Do you remember this way back? About how the sea urchins, when they're in the wild, how they can actually change their shape. You know, so they're really quite, we call it quite plastic in their nature, how adaptable they are to living in certain environments. And we were finding that this particular species of sea urchin, you know, in certain environments it could be a lot flatter, longer spines, just was very, very adaptable to life outside of the culture environment. And also Adam took the lead on really trying to see the role that these sea urchins played in the transmission of carbon from those sort of nearshore coastal sort of kelp beds and then into the sort of wider coastal environment. So those two, so it wasn't just looking wholly at the culture, but also looking at these urchins in the wider environment. And not only the wider environment, but looking, predicting what may happen in the future. And hopefully most of you have heard about the changes that are afoot in the acidification of our oceans through climate change. And I had a PhD student, Colleen Suckling, together with some folk down at the British Antarctic Survey, who looked at different types of different levels of pH, the water, and raising those adults, and then seeing how that then affected their larvae that they produced. And as you can see here, they didn't do too well. This one here has what's called a rudiment, and that's the rudiment which is then, when the larvae settles out, it's the rudiment from which the new sea urchin appears. So this one here's got two, and it's kind of missing a limb. And this one here has got some weird sort of growth going on its body. So they weren't doing too well under this reduced sort of pH environment. So, but sort of moving on, I didn't just stick with sea urchins. It kind of expanded the various projects we worked on. So, so one of the ideas was, well, we know that these sea urchins are growing really well on salmon feed. So what happens if we actually put them in a cage with sea urchins, or hang them in nets around those salmon nets so they can benefit from any waste sort of salmon feed or particulate material that came out of those cages. So we did some work with Loch Duet salmon farms, like way up in the northwest corner of Scotland. Good 5-hour drive I used to do every month to get all the way up there, but the scenery, and it's just so fantastic there. So we looked at that, and the urchins did really well growing alongside the salmon. There's a picture of me, oh my gosh, in all my waterproofs on one of the salmon, one of the walkways between the salmon cages. And we also, interestingly, worked with this brilliant character here, Mckinnon, was it? Mckinnon, yeah. Ian Mckinnon. He's so proud that he's grown these beautiful sea urchins alongside his mussel lines. So we also grew them with mussels to see whether they would feed on any of the broken mussels or some of the smaller ones. And I think they did in a way, but I think they also did really well on the fouling that occurred on the netting, on the nets around them. So, so I sort of branched out. And then, boy, I branched out even further. And as part of something called the Atlantic Arc Aquaculture Group, this is a postdoc that, and Maeve was obviously the lead on this, but we worked as ourselves in Scotland, there was Ireland, Wales, France, and Spain. And each partner worked on a different sort of aquaculture species, but it was really nice. I must say, I did enjoy our trips to France, because then I got to eat these incredible oysters that were kind of this size. And we learned all about sort of techniques of how to grow them in these special ponds that they have down there. But I also did some studies on a marine mollusk called an abalone, orma, something like this. They've got beautiful shells, mother of pearl in your earrings. They're absolutely beautiful and they fetch a huge price. In fact, in South Africa, you have to have armed guards on your farm because there's a big black market in abalone. So I did some variety of work, sort of branching out, looking at other different species. But unfortunately, Kenny couldn't be here. I love this picture of him. He looks like he's like doing that dance, sort of walking like an Egyptian or something. So this was taken in Israel. But I also worked on a second project looking at the potential use of a variety of different species that you could hold sort of adjacent to those salmon cages in order to sort of mop up all those nutrients that are being released from the salmon cages, both the particulate and the dissolved nutrients, and we'd see how effective that was. And basically these structures here, that's me there deploying it off the back of our small research vessel, the Sholmara here. But all of these exact same sort of modular units were deployed, so we had them in Scotland, in Greece, in Crete in fact, in Slovenia, just off Piran, and in Israel. So all going in the water all at the same time, and to see what results we got. But this was sort of like the beginnings, I think, of the whole integrated multi-trophic aquaculture sort of scene, although previous work had been done in North America. But I just had to include this, otherwise Rory, my son here, would never forgive me. So I'm very sorry. He's really into Minecraft at the moment. This is a complete divergence. Really into Minecraft. And when I came back from here, he was like, I can do you a salmon farm, mummy, I can do this. So on Minecraft, as you do, this is the farm. These are all the salmon lines. Oh, sorry, seaweed lines. So these are all the seaweed lines that he's done beautifully, and they're just like in Tanzania. All the seaweed lines here, and then just next door you have your sea. This is if you went onto Minecraft, you'd see all the fish swimming around. It's fantastic. So you have your salmon or your finfish here, you have your seaweed here, and then in the ideal world you'd have mussels or sea urchins, and you'd have sea cucumbers underneath. And this is something that Adam at the back here is sort of spearheading, all this integrated multi-trophic aquaculture, okay? So we had these biofouling structures. As you can see, from just the mesh, within 6 months, in certain areas, they became absolutely covered in fouling. There's like you can't really see it, but there's a sea urchin poking out the top there, and tunicates and sponges and seaweeds and everything. And what really became clear to us was, like, you think that if you put your filters really close around your cages, then they would do the best. They would do great because they would be there right on site to suck up all those, like, act as a sponge for all those nutrients being released by the salmon and by the sea bream and sea bass in the Mediterranean. But in fact, it was actually this one here just was such an outlier, and this was in Piran, so in Slovenia, so you're right up north in the Adriatic Sea. And it was really interesting that from this and a lot of other work that's happened since, that you don't have to place your second species right next to your farm. In fact, sometimes it can actually be deleterious to the growth of that species. But dependent on that sort of circulation that occurs in the area, you could well actually put your filters or grow your second species or third species that little bit further away. But you really do have to understand the sort of dynamics, the hydrodynamics of the area. Anyway, okay, Act 2. Act 2, oh my gosh, they're starting to escape. Right, we've cultured them now, but okay, and look what I found on my biofilters. I would literally raise up them, bring them back to the lab, and they would just be crawling at certain times of year with these beautiful beasts here, okay? And I couldn't help it, they're Triffids, they're Triffids in miniature. Okay, they're not plants, but, they're called skeleton shrimps. They're amphipods. They're called skeleton shrimps. Unfortunately, there's not much meat to them, so you can't really sort of have them with your chips. But anyway, they were interesting because we were looking through all the books, all the ID books. We couldn't find anything. We couldn't find them in anywhere. And fortunately, We had Kate Willis here, who was an amphipod specialist. I had an Erasmus student, Matthias. He went through all the books and all the literature, and we contacted a professor in Japan, and it turned out they were native to Japan. And hence my career on my invasives began. So, again, following on, you know, from what we'd done with the urchins, we wanted to know a little, oh, well, yes, urchins a bit later. We wanted to know, okay, right, we've got them here on the fish farm. Sorry, this was literally the fish farm at Dunstan, like there, just the other side of the building. Well, okay, well, where else are they? And I had this fantastic PhD student, Gail Ashton, who came, and she basically did a great tour around the west coast of Scotland. And surprisingly enough, they were everywhere. But the farmers had just been sort of saying, they were causing, they'd get on you and they'd nip you and they were awful. But they hadn't realised that they were not native to here. So Gail actually went and found them all up the west coast of Scotland. And she primarily targeted fish farms because that's where we thought they mainly were to begin with. So that just gives you, so the colour, the black dots are where they were actually found in her study. And then she decided, well, okay, well, we know they're here, so maybe there are other places in the world. And sure enough, sorry, this doesn't come out so well, but they're native to sort of this kind of area, around Japan. But oh boy, they were on the East Coast, they were on the West Coast, they were throughout Europe, and they'd even turned up in New Zealand. So it was really, wow, okay. So we're not so special after all, so there were various other places too. Gail also did some studies looking at just their seasonal patterns. I should say Mike was involved in all of this as well. Remember flooding back Mike. But this is a colleague of mine, Marco Fass in the Netherlands, and he sent this through to me because it just shows that this is an experimental structure that he had in the water in Belgium. And can you see the density? It's just like a mat of them, and Gail was recording them at 300,000 per metre squared. And there are fish farmers that were recording, they would lift up the nets of the fish cage, you know, the nets were quite big. I should say these guys were about 40 millimetres long, and you would lift up the nets and you wouldn't, there was no holes, it was just a writhing mass of these caprellids. So Gail looked at the seasonal patterns, finding, you know, we had this peak, or sort of quite a peak sort of springtime or sort of June time, early summer, and then another one sort of in October. And there's a funny story that Adrian will tell you later about trying to find them at these times of year. Anyway. So not only were we interested, though, in studying where they were and their distribution, but Gail also went into a little bit more detail in actually trying to see and use, it was like becoming like a detective. And that's what I love about this work, like a detective, alien detective, in trying to look at their parts of their genome, so their mitochondrial DNA. And she took samples of caprellids from the native area, from the east and west coasts of America, and all around Europe, and saw, because if they're alike, then the chances are that that's the sort of pathway, that's the jump. Because if you've got something that's similar in the native range and it's mitochondrial DNA, and something similar in a place in Europe, the chances are there was maybe a direct transfer. So she started to sort of try to tease out what the roots were, what the pathways were. And it actually turned out, I think there were quite a few introductions into Europe from the native range. And we potentially think it could have been due to aquaculture, but also hull fouling might have been an issue with this as well. And not only did we start looking further afield and looking at other sort of areas, and this particular study was looking at marinas because they were quite easy to get to and relatively easy to sample. But we thought, well, maybe we could expand and start looking for see if there's any other non-native species out there as well. And this was way back. When was this? Way back in the sort of 2006, I think this was published, but there was really so little known about invasive species at this time. And I was asked to join a sort of a group looking at producing the Great Britain non-native species sort of strategy at the time, because there were so few of us looking at this. And actually we produced a target list and I should say I can't thank Chris Beveridge enough here, and there's Richard Shucksmith and Marlena Jenke that helped me with all of this work. But we actually, pretty much wherever we went, we found some non-native. Caprella musica obviously was in many other places, but this is called wireweed, Japanese wireweed, Sargassum muticum. This looks like a slug, but it's not. It's actually a tunicate in a bowl. But this clubbed, call it the club tunicate, from the Far East, thought to have been introduced on warships coming back from the Korean War. And we also have a sort of spongy green seaweed called Codium fragile, subspecies fragile now. So we were finding every time we were going out, we seemed to be finding more. And it wasn't just these other species that I was working on. We were also looking at the spread of Sargassum muticum, the spread of potential sort of Pacific oysters around the coast. Pacific oysters, very much introduced back in the sort of 1960s, Walter, 60s into Europe. When at a time when the seawater was just too cold for them to reproduce. They just, it was fine, bring them in, great. The native oysters were being plagued by various diseases. This offered an opportunity for the oyster industry to thrive, and it has. But as seawater temperatures have increased, they are now starting to reproduce in the wild, from far more down on the south coast of England, but we were also out doing a survey for them, finding them on the shore in the Solway Firth, the northern shore of the Solway Firth there. And also, look at this. This is, it looks like candle wax or something. It's dripping, isn't it? I mean, this is a colony about that sort of deep. And this is the carpet sea squirt, Didendum vexillum, that really is, when you get it, it can cause huge problems. So this is something that we're working with the aquaculture industry here in Scotland to see if we can try some way of controlling it. So we also, as part of the work on invasives, I led a UK-wide group called Marine Aliens that we got funded from the Esme Fairbairn Foundation. And this was just going back to 2007. So you can see we were starting to sort of populate the map with our key sort of species that we were looking at as part of this group. And as one of the outputs, it was such a wonderful event, but we got to host an event at the Linnaean Society down in London. And this chap here is Dr. Paul Clark from the Natural History Museum, who is a fellow of the Linnaean Society. And we got to speak in this incredible room here with pictures of Charles Darwin and original oil paintings, and Linnaeus and Charles Darwin looking down on us. It was a very grand event. And we also, which was wonderful, got Professor Jim Carlton. He's in the States, and he's like the godfather of invasives research. So it was really wonderful that he could make it to our meeting. But I just wanted to put this in to compare. So this just shows you, if I go back, if you just take a look at what's happened there with the sightings in 2007. And this is another PhD student of mine, Chris Nowell. He then went and did another survey around Scotland and also looked on, it's called the National Biodiversity Network. And he then also collected sightings of invasive species. I think this was published in 2015. So it just shows you that just over that last 10 years, there's been so many more sightings. I think because more people are out looking for them. But anyway, that was part of his work. So I was also involved in, with this fantastic guy here, Dan Minchin, who I'm still working with even today. We not only wanted to sort of also almost collate what information was out there, but do some more analysis, in-depth analysis into sort of the pathways and what types of species are sort of going to be the most problematic. And the previous review by Claire Eno was back in 1997, so we thought, well, it's about time we kind of updated this. So we produced this alien species report for sort of brackish and marine waters. And as you can see, we were looking way back in the literature, back in, what's that, 1850s, late 1800s, and sort of showing how there's been this increase of the number of non-native species found over time. And of course, with the connectiveness of our world now and the globalization of marine traffic, and also, of course, more sampling effort because more people are out looking for them, I can only see sort of that will continue to grow. But we also have, highlighting macroalgae, arthropoda, the sort of crabs and shrimp, the mollusks, the marine snails, and the mollusca and the polychaete worms sort of featuring quite highly there. But with the key thing that, of course, these species are quite large, and it's quite easy to see when something doesn't look quite right. But there's a whole world out there of protozoans and cnidarians and some of the sort of smaller bacterial, the viruses, that we really are only starting to get an insight into, really, and how they're being moved around the world. So interestingly, so we were looking at where they were, where they kind of moved and how they moved. We were also, well, what impact are they having? And this is really hard in the marine environment. In freshwater, I don't know whether many of you have seen pictures of Lake Victoria. There's a particular type of freshwater algae that's sort of covering parts of those lakes in Africa. But in the marine environment, because it's far more open, it's much harder to sort of get a real indication of what effect some of these species are having on our native ones that are living here. And I was lucky enough to team up with Joanna Diaz and others at Aberdeen, looking at developing assays, looking at PCR and DNA assays to see how certain species sort of hybridized with one another. Not necessarily Walter's about Trosylus, Mytilus trosylus, and whether it's actually non-native or not, or whether it was here before. But it was interesting how you can start to use the PCR work and the DNA work to looking at whether potentially a non-native species could start to hybridize with native species. But we were also looking at competition, interactions between Caprella mutica and also two other native species of caprellid. And as you can see here, this is Richard Shucksmith's sort of work. This is just like a flat mesh. They've got hooks on the ends of their legs, so they like to have something to hang on to. So this was just a flat mesh. And Richard, he must have been feeling very devilish at the time. He was going to subject these poor, smaller native species of caprellid, which are like half the size of the Caprella mutica, to interactions with the invasive one. And here you have the first bar where there's no Caprella mutica, no invasives, and you can see they're all quite happily sitting on the mesh. You just have to introduce 5 or 15 of those invasive caprellids, and they literally all flee for their lives. I don't know whether you can say that, but they scarper, and you can see that. Whereas then Richard put in a little bit of sort of textured filamentous. It wasn't living, it was artificial filamentous type algae. And just having those refuges, you can see that there is still a decline in the numbers, but they're not quite as significant as when they don't have any of those refuges at all. But this, of course, was done in the lab. It wasn't done out in the field, so the jury is still out in how significant the effect is of this Caprella mutica on that wider environment. So, interestingly, I wasn't just interested in how these particular species, though, were interacting and distributing themselves around the UK and how they were moving around the UK, but I was also interested in figuring out, well, how are they moving through Europe, potentially? Are they moving through Europe? And one particular group, which is called a bryozoan. It's an animal. This is an animal. It looks like a seaweed, but it's actually an animal. It's made up of thousands of tiny animals here. We found this at a number of our sites in Scotland. But interestingly, it had first appeared in the Venice Lagoon. And then there are reports of it spreading all the way up through Europe and around the UK. So it's also quite interesting in building that body of information of how these species move. And we actually was thinking that's probably on the hulls of boats. So I also, at the time, got involved in a wonderful lady. I couldn't find a picture of her that I wasn't copyrighted, but called Bella Galil. She's part American and part Israeli. And she has worked in the Mediterranean for decades now. And she has been tracking the influx of non-native or invasive species through the Suez Canal ever since it was built. And we're probably talking back in the 1900s here. So she has been tracking the number of species that have been introduced through this canal system. And I should say there is like a northwards, northwesterly current from the Red Sea up through into the Mediterranean. And basically, I don't know how many of you were aware, but they have recently, quite recently, enlarged that Suez Canal. And in places, there's even like 2 lanes which you can go, I suppose, to overtake those slower vessels. But the vessels that are coming through are huge these days. And so as a consequence, the Egyptian government, who runs the Suez Canal, decided that they would have to deepen it and widen it. But they weren't putting it, they have not, even though they know that it is a major pathway for the introduction of species that swim through or come through on the hulls. They have yet to do anything about trying to prevent that spread. But I was involved with Bella and a whole host of other international researchers trying to raise the profile of this in a number of different articles, and I'll tell you a little bit more about something, another initiative I was involved in that particular area. But also talking and thinking about pathways. So we've got like, I've talked about shipping, I've talked about maybe ballast water, possibly aquaculture, but also I've sort of been terming these or calling these superhighways, sort of oceanic superhighways. And did you know that the world's largest offshore wind farm has just opened up in the Irish Sea? Many people aware of that? I was actually on a ferry across to see my father-in-law this summer that left Liverpool, or it's Heysham, and went over to the Isle of Man. And this is the site, this is one of my photos that I took. And this is Scotland, I suppose Galway, would it be Galloway in the background? Oh, I'm not sure anyone got the expert in the front seat.
I would say that's somewhere around about Barrow-in-Furness.
Oh, right, okay, Barrow-in-Furness. But it was just turbines as far as you could see. Until you see it with your own eyes, I would do Google Earth or something. It was unbelievable, the amount of wind turbines that are just in that area in the North Sea. I couldn't believe it. And this is happening, and it's been replicated off the Kent coast as well, throughout the North Sea. This is being replicated throughout Europe, and also they're looking at it in the States. But Rayanne Miller, I don't think Rayanne's here tonight, but Rayanne led a paper that we got published in Frontiers of Ecology and Environment, just looking at the sort of benthic footprint that each of these structures sort of had around it, but also, and Adrian was involved in this as well, but looking at, you know, these particular structures here are not that actually dissimilar to the hull of a boat. Okay, they're static, but they're, you know, artificial structures in the water column. And, you know, we started to think of, you know, we've heard of, I don't know how many of you have heard of the sort of stepping stone theory, where you kind of jump from ship to ship to ship, or from wind turbine to wind turbine. So, you know, really sort of beginning to think, well, a species X here, or my didenum or what have you, probably couldn't actually make it across the North Sea normally, because its larvae only live in the water column for 2 to 3 hours. Oh no, sorry, 2 to 3 days. But you kind of put a whole host of artificial structures in the middle of the Irish Sea, and you're beginning to put in these stepping stones to allow it to spread that bit further. And that's where I think these superhighways, and I'll come back to that, are going to contribute to the spread of invasive species, I think, around our coastlines in the future. But I'm not, you know, as well as being interested in the sort of what is happening here and now, I'm also being interested in, well, what's going to happen with climate change? And you know, this happened with the sea urchins as well, that I've already mentioned, but I've been involved. I don't know how many of you have heard of the Marine Climate Change Impacts Partnership. They produce these really excellent sort of glossy magazines. Every couple of years, Mike, we've been involved in this. Is it every year? All right, okay, annual report cards. So I've probably been involved in about 4 or 5 of these, either as a co-author or then leading the charge, leading the writing of them for the marine non-native species. And behind these glossy mags, of course, there is a full sort of peer-reviewed paper. But this is quite good at just giving you that overview. And so it's not just looking at the here and now, but it's also thinking, well, what's going to be happening in the next 10 years? What's going to happen in the next 20 years? And how is our environment going to be influenced by non-native species? So there's a few of those outside if you want to pick those up. So on to the next part of the story. What's this? Act 3, protective fencing. There's Becky. Do you like this picture? Yeah. So, okay, it all sounds a bit doom and gloom at the moment. We've got this flood of invasive species coming in. They seem to be running amok. Oh my gosh, they're having some impacts, although we don't know exactly. But what are we going to do about it? You know, that's always the question. How can we stop them? What can we do? And this is where this protective fencing came in. Well, what on earth can we do? And this is something that a master's student started, Vicky Foster, but also it's been taken on by Becky now as part of her PhD. And guess what? This is where I come to test you. Okay, so how did they control the Triffids? So how do we control the marine invasives? Ta-da! Okay, 10 out of 10. Okay, fantastic. And yeah, maybe we can't control all of them. This is what Becky's analysing all her data. Maybe we can't control all of them. But we're certainly finding that the nearer your structure is to the river, to a river mouth, then you certainly see a lot less invasive species in that particular area. And Becky's been doing some really excellent work looking at dipping panels into fresh water for certain lengths of time and seeing what particular species get killed off. So we are looking at fresh water in this respect. But we're also looking at putting in, early warning systems, particularly for marine invasives, because the ocean is so open. Is there something, everyone keeps saying in the literature, if you find them quickly, if you find them early enough, it's like Japanese knotweed in your garden. If you find it and you pull it out, or dig it out, then hopefully you've controlled it. But you let it get a grip, you let it form quite a stand, and you take years to get rid of it, and a lot of money. So is there different mechanisms or different types of techniques or innovative sort of new structures or tools that we can use to try to detect these species when they first arrive? And there's been some excellent work done in Australia and New Zealand. A particular type of mussel has been found in a marina, highly invasive. They've only found it in that one marina, and they have gone in, they've shut the marina, they've put gates on the entranceway, and they have flooded it with chemicals to get rid of it. But they've worked really quickly, and we're very poor at doing that in many places here in Europe. But we were looking at early detection systems, and also I did a review not so long ago on looking at all the different types of marine biosecurity techniques that have been out there. There's a great huge marine snail called Rapana venosa. It's got bright orange inside where the meat is. And there's people that have put like bounties on the heads of them. You can get $5 or something for every one that you collect. And there's places in the Caribbean at the moment that they're really being plagued at the moment by the lionfish, which has escaped from aquarium. I don't know, it's poisonous, but you can eat it. And certain chefs are looking, it's got poisonous spines. So as long as you know how to cut it. It's tasty. Oh, good, yeah, I think. So, so they're having cook-offs, bake-offs, cook-offs, or, big cooking events to try to get divers out to catch these lionfish to bring them in. So there's a whole range of various techniques that people are looking at, as well as sort of using freshwater jet washing. So that was part. And I was also part of putting together a guidance document, basically Sarah Brown, who was working at the Clyde Forum here, we worked with her. She was getting fish farmers, she was getting harbour masters, marina operators coming to her and saying, what can we do? How can we stop these creatures, these non-natives coming and settling on our structures? And so as part of a piece of work I did with Sarah and another colleague, Robin Payne, we actually came up with a guidance document that has now been introduced into the Scottish planning sort of legislation, but is also now being used UK-wide by all of the UK environment agencies. And thankfully, this came out just before the Commonwealth Games in Glasgow. And was anyone part of the flotilla that sailed? Hundreds of boats sailed up the Clyde to take part in a big jamboree as part of the Commonwealth Games. Well, they were and these boats were coming from all over Europe to take part in this. And they were absolutely beside themselves with, oh my gosh, one of these boats could well bring in something that's really quite nasty. But they followed this procedure. They made sure a lot of the boats coming from further afield went into, I think, the James Watt dock, which is freshwater. Anyway, fingers crossed, as far as we know, nothing terrible got introduced during those games. So that was a bonus. Is that right, Adrian? Yeah. Oh, sorry, Claire. So another initiative that I took part in that Bella Galil's led as well was, I was mentioning earlier that there's a ridiculous number of invasive species, over 150 now, that have come through from the Red Sea into the eastern part of the Mediterranean. And interestingly enough, around 20% now of the fish species are invasive. It's not just a small amount anymore, but some of the fishermen have actually switched from fishing for Mediterranean species to Red Sea species now. But some of the species are great, okay, they're providing new opportunities for the fishermen, but it just so happens that some of these species coming through have no commercial food value. In a couple of cases, there's a puffer fish that has come through that has caused sickness. I'm not sure whether anyone's died through eating it, but it can make you very, very sick if you eat it. And there are also rabbit fish that have come through. And in the Mediterranean, I'm not sure how many of you are aware, but there's a lot of marine protected areas in the Mediterranean. And these rabbit fish are coming in, I haven't got a before and after picture, and absolutely decimating the seabed. So whatever you've got being protected, well, there's no point in having a protected area anymore because it's gone because of these invasive rabbit fish. So there is a whole host of really, really critical issues that are going on in the Mediterranean at the moment. And as part of this, as part of the Euro Marine Board, that we held a conference, Bella organised it, that I was invited to in Ischia near Naples, and we came up with something called the Ischia Declaration that has been, I don't know what's happened to it, I will tell you in another 5 years, how effective it's been, because it was only out 2 years ago. So we're still looking to see how effective that's actually been. But I'll keep you posted. So we're not far off now. Act 3, how to actually go about conquering those invasives. Well, we've looked at the biosecurity, but also it's not just here in Europe where we have an issue with these non-native species. Of course, we have issues further afield as well, and developing countries in particular have real problems in dealing with these non-native species, because a lot of them have no biosecurity controls whatsoever. They have no border controls. You could literally pick some seaweed, which has happened, from the Philippines, basically put it in your hand luggage or in your whole bag and transport it to Tanzania and plant it out and grow it. And there is absolutely no restrictions on doing that. And this is where Claire, she managed to get some money from NERC, their International Opportunities Fund, to fund a project called Global Seaweed. This is about 4 years, a few years now, to work with seaweed growers, seaweed researchers in various parts around the world, And Claire has done a lot of work on seaweed diseases, and I was brought in because of my biosecurity sort of background and knowledge. And we work really well together and really closely with a whole bunch of researchers in the seaweed industry. I should say here, I would pretty much guarantee that all of you have used a seaweed product today. Anyone like to dispute me? Can if you want. You all brushed your teeth today. I hope you've all brushed your teeth, because seaweed extract is in toothpaste. It's in beer, it's in paper, it's in thickening agents, it's in bread, it's in, you name it, and there is a seaweed derivative in it, pretty much. So it's really massive. It's just that we don't eat it here, but it's eaten. You know, if you're in Korea, if you're in Japan, they eat seaweed like we eat potatoes here. It's like part of their everyday diet, and it's massive. So they have a huge problem though in the seaweed industry, that they're losing up to 20% of their yield each year through disease and pests and pathogens. So this is why we were sort of getting involved here. So this is a policy brief, and again we have a few of them outside that I led with something like 21 other institutes globally, and the United Nations University were really great at helping us spread the word about this, and we sort of had policy recommendations on the background here, on the back page here. But this then went on to us developing, and I think this really played a key role, as well as other publications, of course, but in us getting this money for this project. So this is the Global Seaweed Star Project, as Crichton pointed out, £6 million. Oh my gosh, it's a lot of money, isn't it? £6 million to spend. We work with seaweed growers and researchers in Tanzania, in the Philippines, in Indonesia, but there's also a host of partners also here in the UK, ourselves, we're leading it, CFAS, Natural History Museum, Overseas Development Institute, and then we have other sort of global international organisations on our advisory board. But it really is to look at trying to, one, understand the disease and the pests and the pathogens that are causing such problems in the seaweed industry, then looking about how you can actually manage it through different biosecurity practices, which is where I come in, looking to improve. So maybe we could use more native strains rather than introducing seaweeds from other countries. So looking at improving those strains, that's where Claire comes in, and also to actually empower those seaweed farmers in various ways. So that's where that work came in. But alongside that, I'm also involved in work to do like a horizon scanning exercise. So basically, if you can try to figure out what species are on their way, then maybe you can actually prevent them from arriving. So it's like a horizon. And they do this with diseases and flu viruses and Zika virus and things like that. So looking at that horizon scanning. And along with Helen Roy, who is the sort of key PI based at the Centre of Ecology and Hydrology, and a whole host of other people, we did a horizon scanning for Great Britain. And then we've gone on to do it for the European Union, and that's now in press, in press. European Union. I'm now involved in one looking at the UK overseas territories, which is fab, because this is me in Grand Cayman earlier this year, which was quite nice. And I'm off to St. Helena, I think, next month. And I didn't get to go to the Chagos Islands, which was in August. I think I was on holiday with you guys, so you were very lucky. And then, you know, variety. So, and then we've got a proposal in to do some horizon scanning for invasives in the Arctic. So that's in at the moment, so we don't know whether we're doing that, but that's sort of like part of the whole management system. And where would we be without capacity building and education? And as Crichton mentioned, this is something that I lead. It's an Erasmus Mundus Masters program on aquaculture, environment and society. We call it ACES. And yeah, I mean, this is from the undergraduate days. There's a few faces there you might recognize, through to the first, second and third cohorts. And I'm absolutely delighted we've got some students here from the fourth cohort that have just started last week. So we have them here in the audience. But yeah, it's been an absolute absolutely fantastic experience hosting these students and running this program, even though I've got more grey hairs, I think, than I've ever had before. Yeah, so we, they're taught about the latest tools and technologies within the aquaculture industry globally, but also, I try to, there's a number of lectures on invasives, on biosecurity, and looking at the sustainable management of this global industry. So these are also the team, the Global Seaweed Star team at the moment. And again, we've built into that project the training of young researchers. So we have young researchers from Tanzania, from Indonesia, and the Philippines. That's us all out in Tanzania on a seaweed farm this summer. So yeah. Just to finish, I can't, as part of this talk, I meant to sort of give my vision of how this particular sphere of science is potentially going to go, what direction it's going to go. Maybe there are people in the audience here that are thinking of PhDs in the future, or maybe you're doing it now, or where do I think the work still needs to be done? And I did it in a countdown. I didn't quite have a drum roll, but I've got 3, 2, 1. Okay, so in third position. I don't know quite why I put it like this, but in third position, basically, there is no doubt that aquaculture production, in order to feed our growing population, aquaculture is going to have to grow. Wild capture fisheries have basically stagnated. They're not going anywhere. They're not going to get any larger. But the demand for food and for protein supplies is just going to keep on increasing. Okay? Now, a lot of aquaculture, I can't stress so much, aquaculture is based on introduced species. So much. There is salmon being grown in Chile. You do not have any natural populations of salmon or trout in Chile. And it's the second largest producer of salmon in the world. So aquaculture is absolutely dependent on introduced species. And they are being, this particular species here, Capophycus, so it comes from the Philippines and the Indonesian region, but it's been introduced to over 30 countries worldwide. 30 countries worldwide. So aquaculture production is going to be reliant on introduced species, but surely there must be ways to make it, to safeguard it so these species don't then spread and escape and cause havoc in the natural environment. So something. Number 2, I've already mentioned these before, these global superhighways. We have here the Chinese mitten crab, set up in its city over here, one particular spot, thinking, oh, well, my home, 50 kilometres, nah, I'm not going to be able to travel that far. 30 kilometres, oh, well, I've got a few structures here that I could maybe step across from. Getting there. 10 kilometres, yeah, no problem. I'll set up home on that wind turbine or what have you, and then use it to spread. So I do think that the increase in artificial structures in our marine environment is a key aspect of this particular subject area that needs more work done on it. And number one, drum roll, number 1. So I've just said is the Arctic corridor and the opening up of the Arctic with the decrease in the sea ice content, is that going to be the next Suez Canal? I know it's not a man-made structure, it's not a canal system, but we are now opening up new pathways. There are vessels that can get now, if they use this Arctic corridor, if they use these Arctic corridors, I heard a figure with vessels basically going from the Far East through to Europe. Instead of having to go all the way around the south and through the Suez Canal, it takes 15 days off their voyage time. So 15 less days for something to cling onto a hull, or to survive in a ballast tank, or to be exposed to any other sort of forms of pollution or environmental changes. So I really do think that we need to look at this in more detail. Oh no, I spoiled my thing. And actually, it's. I am hopefully going to be submitting a grant in February to the European Union about to look at this very topic and look at how we believe the influence or how we believe invasives could have an impact on marine biodiversity in this particular area over the next sort of 25, 50 years. So hopefully we can start studying this now. But last two slides. So just to finish off, I can't say thank you enough to my parents, to my husband Finlo, to my boys, stop reading, to my boys, for all their support. Everyone at SAMS, you're like a huge family to me. I've sort of been born, I feel like I've born and bred in through SAMS, you know, I've been through the entire system really. To SAMS, to UHI, particularly with all their help through ACES, to my colleagues, I've put unofficial mentors because I don't think they even realised that they were my mentors, and I've got two of them sitting in this room now. So I don't think I'd be here, and my teaching wouldn't be as amazing as it is without Sheen, and my research without me. And of course, I don't know how many of you reckon Angela Hatton here, drinking her cocktail. She's now the Director of Research at the National Oceanographic Centre, so she's done so well. So yes, these wonderful unofficial mentors of mine, all my friends, both present and departed, and of course all my wonderful PhD and ACES students, and of course all of you. Thank you for listening, and that's just some credits. This is just, you know, I'm sure there's people I've missed out, but just all of these people have just contributed to my work over the years, and I really can't thank everyone enough, so thank you.
Thank you for a wonderful talk, Liv.